Hello everyone, my name is Kwon from Mi-Hye University, who is the first author of this article. Corresponding author is Jiyeon Liu. It's my pleasure to introduce our study of gene expression profiling of mouse conus endothelial cells for diagnostic targets in diabetes-induced erectile dysfunction. As we know, the erectile dysfunction is highly age-dependent disorder. Recently, studies suggest that the approximately 5 to 35% of men aged 40 to 70 have experienced different levels of ED. The currently available drugs, such as the PDE5 inhibitors, have shown poor responsiveness in patients with severe endotheliopathy. However, the ET-associated genetic mechanisms in mouse conus endothelial cells from the diabetes-induced ED mice remain unclear in vitro, and more new molecular target responsible for diabetic-induced ED are needed. The purpose of this study is to investigate the potential target genes associated with the diabetic conditions in mouse conus endothelial cells for diabetics-induced erectile dysfunction treatment. For the material and methods, we isolate the mouse conus endothelial cells with material gels and expose to the glucose conditions to mimic the diabetic conditions. Then the RNA sequencing assay was performed. Finally, we used the RT-PCR to validate the new selected target genes. This is the representative images of the spongy cells from the mouse conus endothelial cell tissues at the passage 0 and passage 1. Immunofluorescent staining of the spongy cells also showed the positive staining for the CD31, but not for the other vascular markers. To evaluate the function of the cultured mouse conus endothelial cells, we treated the cells with normal and high glucose conditions to mimic the diabetic condition. We found the mouse conus endothelial cells showed significantly decreased tube formation and migration under the high glucose condition. Next, we exposed the mouse conus endothelial cells with normal glucose and high glucose conditions for 72 hours and harvested the cells and performed the RNA sequencing assay to evaluate the genes expression profiling of the mouse EC in glucose conditions. We found about 23,000 genes in both libraries. Among these genes, about 2,094 genes were upregulated and 16,049 genes was downregulated. From the 14 gene ontology categories analysis of the RNA sequencing data, we found the aging and the angiogenesis have the highest proportion. Therefore, we validated these genes from the category aging and angiogenesis. Finally, we validated these 7 aging-related genes was increased in high glucose conditions compared with the normal glucose condition, and 6 angiogenesis-related genes was decreased in high glucose condition compared with the normal glucose conditions. So in this study, we provided the differentially expressed genes of the mouse conus endothelial cells in normal glucose and high glucose conditions. 7 aging-related genes and 6 angiogenesis-related genes were significantly changed in the high glucose conditions compared with the normal glucose conditions. To study these genes will help us to understand the potential mechanisms for the diabetic-induced ED. Thanks for your attention.